I work at XOMA. So XOMA is a company in the Bay Area, San Francisco. It's a therapeutic company, so it's an antibody company. So we have XOMA has interest in inflammatory, autoimmune, infectious, and oncological disease area. We do have some drugs in the clinic that, as well as some program for the biodefense. XOMA has multiple revenue streams coming from royalties for drugs like Cimzia, and also from collaboration, as well as I mentioned, the biodefense contract. So we do have at XOMA a very comprehensive antibody platform that allows us to find antibody against different targets. So at XOMA, this is the typical funnel for discovery, for antibody discovery at XOMA. We usually start at screening, as a primary screen, multiple or more than 1,000 different antibodies, and then for the secondary screen we go for less number after initial characterization, and then the best clone go for functional screens, and then the in vivo POC assays. We always like to, at XOMA, if possible, to be able to identify functional antibody very early, even at the primary screen levels. And this is why, for example, today I will show you some example how we use the SPR and also the multiplex system, which is in this case the ProteOn instrument, to find a particular mechanism of action very early during the primary screens efforts. Just give you a brief overview what SPR. I'm sure everybody is familiar what SPR is. So it stands for surface plasmon resonance. So this usually you have your interaction protein. One of them, the ligand, is bound to the chip, and then you inject the second protein, in this case antibodies, and then you measure the interaction between these two partners using the SPR system. This is a typical sensorgram that people are familiar with, which consists of a baseline. Then you have an initial phase of binding called association, and then when there is a dissociation after the end of the injection. And typically, people using SPR use regeneration condition to regenerate the surface to the baseline. As I mentioned, I will show you the data from the ProteOn instrument. We have several instruments at XOMA, and we typically make a choice very early at what type of instrument we will use, depending on what question we're trying to address. And for example, here for multi-channel SPR system, this instrument can allow for different things. For example, this is just a few examples. For example, you can test for target binding, or you can, for example, measure the concentration of your antibodies in the soup, or you can also use it to determine the mechanism of action, or you can also test for specificity. So this system actually has the quality of high-level SPR biosensing capabilities, and allow for high to mid-throughput multiplexing capabilities. I'm not sure if everybody has seen how this system works. It's a typical SPR setup. The only difference here is that instead of one flow channel or four flow channel, as you can for example, some of the Biocoris instruments have this instrument have the advantage of offering 6 different flow channels. So and also offer the possibility to orient or to turn the reading sensor 90 degrees, and this will allow you actually to read or to inject multiple or 6 different analytes in the other orientation. And presumably, if you set up your experiment right, you can be able actually to measure up to 36 interactions in the same time, as you can see here from this slide. So today I'm giving you two case studies. So the first case, it's a ligand blocking assay. So the purpose is to develop, as I said very early on during our screening campaign, to identify antibodies that will block the receptor or the ligand binding. So the antibodies are made against the receptor. So we'll be looking at antibodies that will block the ligand binding. The rationale is that because the antibodies that I'll be showing you today are coming from phage libraries, and usually those antibodies are either as a single chain or a Fab fragment, and they are usually coming from bacteria from periplasmic extract. And these antibodies have a high level of endotoxin, and sometimes the concentration of body can be low, and the number of clones can be very high. So this type of antibody doesn't allow to use it for example, for cell-based assay to try to see if this antibody block functional assay. This is why we need to use an SPR system that actually will not be influenced by the high level of endotoxin. Sorry. So the next slide explain the setup that we use to study that. So as you see here in the bottom, so what we have done, we immobilize the ligand at high density using a typical amine coupling system. Then we be looking at the binding of the receptor plus minus antibodies. So what we do, we incubate, for example, our PPE or periplasmic extract with our receptor. Then we inject those antibodies with the receptor and see, for example, from here, this is an example, you expect that a non-blocking antibody will bind to the surface because of the mass increase. You will see an increase in the signal, or if your receptor is a blocker, in this case, you will see little or no binding. So this is an easy assay that allow you very quickly to say if you have potential receptor blockers or not. This is just what I was explaining. So, for example, this antibody being able to bind at the ligand binding site doesn't allow the receptor to bind to the ligand, and then it will be washed out. And that's what you see, for example, from this type of histogram. So as I said, we used ProteOn to do that. So the first thing we did, we injected full concentration of the receptor over surface that has the ligand. As you see here, there is a very nice signal detected, and this signal, of course, dissociates after a few minutes. So we decided to do the measurement to choose a report point at 5 seconds after the end of the injection, and look at this the signal at this point when we add the different antibodies. And this is just an example of this assay. So we used here a known blocking antibody. And as you can see here, when you inject your receptor with this blocking antibody, the signal you are getting actually decrease. So you go from 600 RUs to or response unit to 60 RUs. So there is a good dynamic range to try to see if we can detect any blocking antibodies. So this assay, this is a 96 well plate. As you can see here, this is the data coming from the protein, represented as a percentage of neutralization. So you see we have two positive controls here in those two wells, and you see those two antibodies are able to block more than 50% of the signal. The negative controls don't change or don't decrease the binding of the receptor. And as you can see here, those yellow wells, there is a potential of antibodies that block the signal more than 50%. One thing we do, and the protein is very helpful, is it's very important that we determine how much antibody is in the spirit-plasmic extract. Sometimes the concentration of the antibodies in this plasmic is very so you can get up to 10 micrograms, but you can get even less than that. So it will be important to detect how much antibodies you are injecting. And this is something you can do in parallel when you are doing your receptor blocking assay. So basically you can, for example, in this case, immobilize and multi-his antibody that will allow you to detect the single chains in this periplasmic extract. And as you can see here, there is some antibody that actually express lower than others, and those presumably they will not block the ligand, the receptor, and they will appear as positive as ligand blockers or receptor blockers. So this slide actually showed a screen that we've done with different antibodies, and as you can see here from here, a lot of antibodies actually are able to bind and not to block the ligand. But we do also have some potential blockers here. We have 3 antibodies that showed some decrease in the receptor binding, and particularly this clone 153 actually was showed to be very strong blocker. So there is other options, or there is alternate formats to, for example, to do these assays. The first assay have some pitfalls, and I'll explain to them in a few minutes. The alternate format I'm showing here, this is actually similar to the other one, but it just the first what you do is you immobilize your ligand, then you inject your receptor, and then as a second injection you inject your antibodies. So for a non-blocking IgG, you will expect to see a significant binding to receptor, and then an antibody that interfere with ligand blocking will not be able to bind. So we run this assay for this same assay with this alternate format, and we did see actually different results, but we also found that some antibodies actually we're showing blocking in both cases, and particularly this antibody 153 that's also blocking this assay. So as I mentioned, the type of configuration you are using to do this experiment may affect the result, and this is due to several reasons. One of them you have to always pay attention to is, as I said earlier, the concentration of the antibodies in your supernatant, and also, if you have an antibody with low affinity, this antibody will be also may show as a false positive. And particularly for the first, for example, for the first assay, if you have a low affinity or low concentration, as I said, the antibody may look as a non-blocker. But for the second assay, for example, the low affinity or low concentration will look like a blocker. So when you run this type of assay, you have always to have your controls. You have to be able to detect how much you have in the soup, and confirm the result with the secondary screen to really to say if these positives are real positive or just false. One thing actually that the protein is very good at helping our effort is what's called binning. So basically, you try to bin different antibodies, see if antibodies actually share the same epitope or they are not overlapping. And for example, if a typical antibody has a particular epitope, you would expect the antibodies that bind the same epitope, they may have the same mechanism of action. And the rationale is that you can actually also very early, by using a known blocking antibody that you have, to try to find a similar antibody with the same characteristics. And this is actually represented here in the slide, as you can see here. So what we did, so we immobilized two different antibodies. One of them is a ligand blocker, and one of them is not ligand blockers. Then we screened for antibodies that will pair or not with this antibody. So if an antibody doesn't bind in the presence of the first antibody, you may say that these two antibodies might be overlapping and or affect each other with a different mechanism of action, like allosteric mechanism or non-direct competition. So this table, for example, it showed that some results. So we were able to pair or to determine the epitope or the bin this antibody to get two different antibodies. One is a positive and one is a negative, and we're able to find, for example, the one actually that showed. The one actually that shared the same epitope have the highest blocking activity by SPR. So for the second example I'm going to be showing today, this is I showed you a system where that consists of only one receptor and one ligand. So this is that's something that is it's very very easy. A lot of receptor I like that, but not all of receptor I like that. There is a situation or the binding, or for example the signal involves multiple components. And in this case, I'm going to be showing a result from this receptor called IL-1 receptor. So this is a cytokine receptor. We are interested to this receptor because we have in the clinic an antibody that targets this ligand iron beta. So IL beta is known to be involved in different disease area, and blocking this ligand may be very beneficial for patients that express high level of this cytokine in their tissues. So this receptor, actually, a lot of work has been done to try to understand how this receptor acts and how the signaling occur. So the first thing that happened is the ligand bind the receptor in the left, and this induce a conformational change that actually allow an accessory protein called the RACP to bind, and only after this three molecular complex happen that the signaling pathway starts. So as you can imagine, if you are interested in studying this system because of this multiple component interactions, to determine kinetics of these multi-components is very challenging. So you have to try to isolate this individual interaction while you are retaining, of course, the activity of each component. And in addition, you have to study this interaction plus minus your antibodies. So typically, this is a very complex situation, and the SPR system and the multiplexing system like the ProteOn actually help us to understand or to depict the kinetics on the interaction of all these components. So this slide showed, for example, what's called Bio-Rad or the ProteOn called the one-shot kinetics. So what we have here, we immobilize the receptor, so the soluble receptor R1 in the chip by Biotinylation. Then we inject fixed concentration of the ligand, and this is actually show you a nice ligand binding to the receptor, and this is you can do it in one injection. You can get a very good estimate of the affinity. That here is subnanomolar affinity. And one thing actually what we did also, we looked at the binding of the receptor or the accessory protein. As I mentioned earlier, the accessory protein actually is able to bind only upon ligand binding. This is the initial injection showed the I1 beta binding to receptor that will dissociate after. So we use the co-inject mode. This is a feature from the ProteOn as well as other instruments. So you can inject actually two ligand or two molecules consecutively. In this case, so we inject the ligand, and then we inject, after that we inject the accessory protein. As you can see, the accessory protein is able to bind when the ligand is bound to the receptor. I'm not showing the data here, but the accessory protein actually is not able to bind without the ligand. So the presence of the ligand is very necessary to the RACP to be able to bind. So as you can imagine, studying the kinetic of this interaction can be very straightforward, but when you start dealing with three different molecules at the same time, you will get a complex kinetic because of the dissociation that you are getting from here. This is a mixed dissociation between dissociation of RACP and dissociation of IR beta. So the kinetics here can be, or the off rate, at least the off rate part can be very challenging to determine. So we're able to try to study that by using different methods. So one of them here I'm showing you here. So for example, what we did here, actually we injected, we immobilized the accessory protein, the chip. Then we injected the receptor plus ligand. So we added the ligand in excess of the receptor. So all the receptor is binding sites are occupied, and then we can actually determine the association of this complex to the ACP, and also we can detect the dissociation of the receptor from the ACP by adding the ligand only in the dissociation phase. So this is not a perfect system, but allows you actually to separate those complexes and try to get some fair or some rough estimate of this tri-molecular interaction. So coming back to the ligand binding assay that I just showed you here in this sensorgram, to be able to actually to determine accurately the affinity. So what we have used what's called global analysis. The protein, as I said, offered the possibility to immobilize the receptor at different densities here. So we immobilized the receptor at different densities. Then we injected the ligand, 6 concentration of the ligand in the same time. And in this case, you can get kinetics from this for example, 4 densities of receptor. As you can imagine, when you're, it is known in the SPR field that when you have a high density receptor in your chip, you will have issues with the mass transport and also with rebinding effect. And as you can see here, when your density goes up, this part of the curve actually has it appears slower than that's what you see here. So the global analysis actually allow you to avoid this type of problems. And as you can see from the residuals, so you see that at low density you have a good fit by when you fit this interaction to mass transport limited model. So our interaction actually is that mass transport limited. The ligand bind the receptor with very high. The on rate is very fast, and we couldn't avoid to have mass transport even at low densities of receptor. So this is what I was referring to. So the slow dissociation that you see could be explained by mass transport or rebinding effect of the ligand. And as you can see, there is a fast phase and there is a slow phase. So the question we ask is, does it really this slow dissociation is really due to mass transport only, or there is other factor that actually give this type of results? And one of the models that we were looking at is this receptor is known to have as I mentioned earlier, when the ligand bind this receptor trigger the ligand trigger a conformational change in the receptor that allows the receptor to close, and this change, that conformation change that occur that allow the accessory protein to bind. So it could be that this slow phase is due to this type of event that happened. It is really it is known in the SPR field that SPR is not sensitive or enough to detect conformational change just because of the this event happened very, very quick, and it's very difficult to see them, or the SPR is not sensitive enough to detect conformational changes. This is the structure. There's several structure from this receptor, as I said. This is the receptor without ligand, as you see here. This is a very compacted structure. But we see when you have the ligand bound to this receptor, the shape of the receptor is completely different. So it's a very open conformation, and in this case, it's very close conformation. There is another molecule called RA, which is an antagonist. So when it binds the receptor, actually blocks the receptor from signaling. And actually, this antagonist actually is not able to trigger the accessory protein binding. And the explanation from at least from the structural studies that was done is that this antagonist is not able to trigger or to bind the second site from in the receptor. In contrast to A1 beta, which is able to bind at site 1 and site 2. So there is out there evidence, either by chemical evidence or structural evidence, that suggests the receptor might have multiple conformation stage, and the ligand might induce these transitions. So one step we try to do and try to understand this phenomenon. So we use what we call directed immobilization. So basically, what we do, we use biotin that is linked to a flexible linker. So we do biotinylate all the receptor or our co-receptor by this biotin linker, and then what we do, we form a complex. So we take those biotinylated proteins, and we add the ligand to them. So we add excess of ligand. So both protein will make a complex. Then we immobilize these complexes over an SPR chip. In this case, a GLN chip. Protein GLN chip that has neutrophilin. So this complex will bind to the GLN chip, and the one that not binding it will be just washed out. Just by adding the buffer, actually the ligand, because it's not binding it, the ligand will be released, and then the accessory protein and the receptor will stay directly or captured by the neutrophilin. And in this case, you will be able actually to study the kinetic of the ligand to receptor, and you can also study the effect of the accessory protein to this interaction. So if you inject the ligand, you get association, and then you can also have data on dissociation. So and one thing that we tried also to address is when we do that, we try to add an excess of co-receptor just to make sure, because the affinity of the accessory protein to the receptor is very it's in the double-digit nanomolar. So we had to add an excess of co-receptor to ensure that most of the receptor is saturated or complexed with the accessory protein. And this is what I will show you in a minute. So we use the SPR, the protein stream to do that. So that's what we did. So we injected several ratios from receptor and accessory protein. So the first flow channel, so we use only the receptor. Then we start increasing the amount of fold ratios of accessory protein, and we have control with just accessory protein. So then in the other orientation, we can inject actually 6 different concentration of iron beta. Then we can determine simultaneously the kinetics from binding of iron beta to 6 or 5 different immobilization conditions. And this is what you see here. So in this case, this is a receptor. This is what I've showed you before, receptor only. So the receptor bind and to dissociate. We still do see these 2 phases of binding. But when you have an increased amount of accessory protein there, you see that the off rate gets slower. So we get even very slow off rate when you have high concentration of ACP. We could fit this the first time. We could fit this with the mass transport model to try to to avoid heterogeneity that we see in the dissociation phase. But when you have the accessory protein present, we will be able to fit this interaction only with the simple one-to-one lentimer binding model, as you can see here. And this, as you can see it from the residual. So when you have high concentration of ACP, actually this interaction became, as we are looking at one interaction. So it's very homogeneous. There is no heterogeneity, and the residuals are nicely. The fit to the sensorgram is very nicely nicely done. So from this result, what this suggests to us is that in the case where we have only the receptor, that the ligand bind the receptor may be able to bind the open or a closed conformation, and this transition between open and closed conformation may be responsible of this slow dissociation that we have seen. We can fit this interaction by using either heterogeneous ligand binding model or using a one-to-one model that include the mass transport parameter. But when you have a receptor that actually is complexed with accessory protein, or it's immobilized in presence of accessory protein, actually this interaction became more homogeneous, and you can actually fit this interaction with one-to-one model. Something that I have to say is that when you immobilize your receptor or your receptor accessory protein without the ligand, you don't see the type of stabilization we see in the off-rays. So having the ligand when you put these two molecules in the chip is very important. So the ligand allow the receptor and accessory to be oriented properly in the chip, and that's what we see. We see that at high concentration of ACP, actually the ACP is able to stabilize the binding of the ligand, and the off rate gets even slower. So our conclusion is that when you have this type of receptor, that the ligand, when it binds these two sites in the receptor, actually the kinetics are influenced by this type of transition between the open and closed conformation. And when you have the accessory protein present, actually there is a preference for the closed conformation, and we see it from the kinetics that we obtain. This is a schematic here. Just to confirm what we are trying to say, so what we did, actually, we tried to generate some mutant. In this case, we generate a mutant called K93A. So this mutant actually affects the binding of IL-beta to receptor. So there is a decrease in the affinity. There is a 50-fold affinity decrease, and this is mostly due to on-rate effect. So you are affecting the on-rate. So the reason of doing this is we try to decrease the on-rate to see if the slow dissociation we have in our curve is due to mass transport and to the fast on-rate that this cytokine have. So by decreasing the on-rate of this ligand, we use the same system I showed you before to see if the receptor if this mutant can be stabilized in presence of accessory protein. And as you can see from the left, this mutant is compared to wild type or another mutant, it has low affinity to the receptor, But when you have this receptor in presence of accessory protein, actually this this mutant is still able to be stabilized by the ACP, which suggests to us that the stabilization we have seen in our dissociation is not only due to mass transport, but there is also effect of the open and closed conformation from the receptor. So in summary, so here we determined the kinetic of a very fast on-rate ligand binding interaction using a fit that incorporates mass transport parameter. So we can actually isolate all these interaction by using specific formats, and the one-shot kinetic actually helps a lot to understand or to study this very efficiently and in a timely manner. So the accessory protein binding and ligand stabilization actually suggests that the receptor have an open and closed form that we can actually see from the kinetics we have, and these kinetics actually or this type of kinetic actually will help us actually to understand our both our target and also to understand our antibody mechanism of action. And finally, the SPR platform actually that offer this type of flexibility, like the ProteOn, actually helps a lot to increase the throughput and also help to enable us to continuous innovation and also adapt novel assays to support all our phases of antibody discovery. Thank you, and we'll take some questions.